So, ladies and gentlemen, you are all welcome to our today's class. So, in today's class, we are going to look at the carbohydrates chemistry, that is chemistry of carbohydrate. So, let me share my slide. So, once you see my slides, if it's actually show, then you now indicate by saying yes. So, can you all see my slide now? Can you see my slides? If you can see the slides, indicate by saying yes. So, as I said, today we are going to look at the concept of carbohydrate or chemistry of carbohydrate. So, what is carbohydrate? First, we need to understand where do we actually get carbohydrate. So, in nature, carbohydrate is widely distributed, and we can actually found carbohydrate both in plant and animals. Although carbohydrate is found in every living organisms. It's found in every living organisms because we know that from the basic knowledge of biology, the current organisms or the classification of organisms now is beyond plant and animals. So, but you can get carbohydrate both in plants. You can also get it in animals. You can get it also from microorganisms. You can also get it from algae. So it is everywhere. So each and every living organisms on earth possess carbohydrate, and this carbohydrate have a variety of functions, and we are going to look at the functions as well as the distribution of carbohydrates in every living organisms. So now carbohydrate is a major source of energy from our diet. So carbohydrate is actually used in energy generation in the body. Although it's not only carbohydrate, we have so many other classes of macromolecules. Like for example, apart from carbohydrate, we have lipids, we have proteins, we have vitamins. They are all class of foods and they all generate energy. Carbohydrate generate energy, lipid also generate energy, as well as the protein itself. And we need to understand that the energy that we are getting or that we are generating in our body is what makes us to do a lot of work. Like for example, now if you are reading is because of the energy that you obtain from your diet. If you are doing anything, any physical activities that you are doing, the reading, if you are doing anything that you know is because of the energy, and most of this energy are generated from carbohydrate. Although, as I said, other class of food also generate energy. In fact, the energy generated by the lipids is even twice more greater than that of the amount of energy that are generated in carbohydrates. The only thing that is that the carbohydrate is an immediate source of energy in the body. So once you need energy, the immediate source of energy in the body is carbohydrate. So this carbohydrate is made of the three major elements. There are three major elements that are found in carbohydrates, and these three major elements include carbon, hydrogen, and oxygen. And of course, the ratio of carbon to hydrogen, so or hydrogen to oxygen is is in a ratio of 2:1. So that is the ratio. And then the next thing that we are going to look at after this is the definition of carbohydrate. So, ladies and gentlemen, if we are to define or if we are to give the chemical definition of carbohydrate, carbohydrate is actually defined as a fully hydroxyaldehyde or ketones or any compound that yield this on hydrolysis. So it means that if you want to define carbohydrates, there is a basic is because generally so many people, if you ask them the definition of carbohydrate, they will start telling you that carbohydrate is an energy giving food or carbohydrates is made up of carbon and hydrogen. And truly speaking, that is not the definition of carbohydrate. The definition of carbohydrate, because this is carbohydrate chemistry. So if we ask you to define carbohydrate, so carbohydrate is a fully hydroxyaldehyde or ketones or compound that yield these on hydrolysis. So what I want us to understand here is this. In organic chemistry, if you did organic chemistry, we have what they call a functional group. So any compound that called or that considered as a carbohydrate, it must have either aldehydes or ketone functional group. So they must have either aldehydes or ketones functional group. And of course, from our basic knowledge of chemistry, when we said aldehyde functional group, it means that they are compounds that have carbon to oxygen double bond with free hydrogen attached to it. And of course, look at it here. We can see clearly here. This is CH. We can clearly see it from here. This is carbon to oxygen double bond, and there is a free hydrogen. So of course, this is the aldehyde. So for the ketones, there is carbon to oxygen double bond and two different alkyl groups attached to it. So that is the aldehyde. So the fully hydroxyaldehyde or fully hydroxyketones is what we considered as a carbohydrate. So it means that one, the criteria for a particular compound to be considered as a carbohydrate, the first thing, the compound must have either aldehyde or ketone functional group. So you should understand that for the compound to be called or to be considered as a carbohydrate, there are two set of criteria that you shouldn't forget about it. Number one, the compound must have aldehyde or ketone functional group. And the second one, there must be fully hydroxy. When you say fully hydroxy, it means that there must be so many OH. Like for example, if you look at the structure of the aldehyde here, or maybe glyceraldehyde, look at it very well. You can see clearly that there are two OH groups. So that indicate that this compound A is actually a carbohydrate because number one, it has aldehyde functional group and there are two OH groups. So carbohydrate is a fully hydroxyaldehyde or fully hydroxyketone. So that is why even in the case of ketone, you can see that there is oxygen double bond with two OH group. So that is carbohydrate. And there is also a general formula for carbohydrate. Carbohydrate have a general formula. And the general formula for carbohydrate is CN open bracket H2O close bracket N. So this is the general formula for carbohydrates. And so also carbohydrate, it is also called saccharides. It is also called saccharides. And we have examples of carbohydrates. These examples of carbohydrates, we have C6H12O6, which is glucose, and then we have sucrose, which is C12H22 or 11. And generally, the carbohydrates that have aldehyde functional group, they are called aldose. So all carbohydrates that have aldehyde functional group, they are called aldose. And then those carbohydrates that have ketone functional group, they are called ketones. So, ladies and gentlemen, in this slide, we were able to define carbohydrates. That carbohydrate is a fully hydroxyaldehyde or fully hydroxyketones or any compound that produces such on hydrolysis. And of course, from the image there, we can clearly see that we have aldehyde functional group and we have ketone functional group. So for the aldehyde functional group, there must be a free hydrogen attached to the carbonyl carbon. And then for the ketone, it is actually another alkyl group. So two different alkyl groups attached to the carbonyl carbon in the case of ketone functional group. So, and then the next thing now is the real structure of the compound. So, if you can see clearly here, we can understand that at this point we have look at the aldehyde. So, for the aldehyde, there is carbon attached to a free oxygen and then free hydrogen, and that is aldehyde. And of course, if you check at it, if you look at it, you see that there are so many OH, that is hydroxyl group. So it's a fully hydroxyl group with aldehyde functional group, and then we also have ketone functional group, which is fully hydroxyketones or fully hydroxyaldehyde. So it means that carbohydrate is a fully hydroxyketone or fully hydroxyaldehyde, which can be clearly seen from the structure here. So you can see that clearly from here there is an aldehyde functional group and there is OH group. So carbohydrate is a fully hydroxyaldehyde or fully hydroxyketone. So what are the major functions of carbohydrates? What is the major functions of carbohydrates? So number one, the major sources of energy in the body. So carbohydrate is considered as a major sources of energy in the body, and of course we have brain cells and the red blood cell. So red blood cells and red blood cells are almost wholly dependent on carbohydrate as an energy sources. So these are the two cells of the body. Although it's not only these the eyes, the rods and cocks and corn cells of the eyes, they are cells that are found in the eyes. So including brain cells and the red blood cells, they depend only on glucose for their energy generation. And these are the reasons. The red blood cell, the rod and conch cells of the eyes, they are the cell that doesn't have mitochondria. So any cell that doesn't have mitochondria, so most of those cells they only depend on glucose for energy generation, or they only depend on carbohydrate for them to generate energy. And the reason why brain also use carbohydrate as a major source of energy is because brain require a lot of oxygen. So fatty acids require a lot of oxygen for it to break down to generate energy. So as a result of that, as a result of that, if a fatty acid or lipids is utilized by the brain, it means that the oxygen that is needed for the proper growth and development of the brain will be channeled to the lipid metabolism, which at the end may lead to actually the hypoxic condition of the brain. So therefore, that is why the brain is solely depend on glucose for the energy generation. So the brain cells, red blood cells, the rod and conch cells of the human body are the cells that actually depend on the glucose for or carbohydrate for the energy generation. And they are also a storage form of energy. So like generally if we eat and we generate a lot of energy in our body, so if there are extra glucose or extra carbohydrate that are not actually or that are not needed to further generate energy in the body, so they need to be stored. So that is why the liver, like for example in human being, if there is an extra glucose that the body doesn't need it to generate energy, so that glucose must be transferred. It must be transferred to the liver. And then the liver will convert the glucose into glycogen. So that is where the storage form of energy in the body is in form of starch in plant and glycogen in animal. So this is to tell you that whenever we take any diet, a lot of energy is generated, and those energy that are generated, if it's actually provides the body with a lot So, what I want us to understand here is that when we take carbohydrates, so the excess carbohydrate that is generated in the body, so it will be actually transferred to the liver and stored in the form of glycogen. And of course, that is what actually happens similar in plant. So plant is in plant, starch is the storage form of the energy. So, and in addition, excess carbohydrates is also converted into fat. So we have when we have carbohydrates in our body, and of course after storage as in the form of the glycogen, so the remaining glucose might also be, or the remaining carbohydrate can also be transported to the adipose tissue and stored it in the form of fats. So then another functions of these carbohydrates, we have what you glycoproteins and glycolipids, which are the component of cell membrane and the receptors. So remember, in the cell membrane, in the cell membrane, there are biomolecules that are usually found, which can be carbohydrate, proteins, lipids, glycolipid, and glycoproteins. So these glycoproteins and glycolipids, of course, when you said glycoproteins, it means that it's a combination of sugar and proteins. That is why it's called glyco. The word glyco stands for sugar. And then we have glycoproteins. So when we said glycoproteins, it means it's a combination of sugar and the lipid. And of course, these glycoproteins and the glycolipid are the component of the cell membrane. So they are part of the cell membrane. And of course, remember that the major function of the cell membrane is selective permeability. It controls what gets into the cell and gets out of the cell. So it's like it controls what will be get into the cell and get out of the cell. And of course, these glycoproteins and glycolipid, they can also serve as the receptors. So when we say receptors, it means that there are biomolecules that are usually found on the surface of the cell. And the major function of the receptors is to bind ligand and send message to a cell for a particular physiological activities to take place. So then we have also the structural basis of many, it serves as a structural basis of many organisms. Like for example, in plants, remember in plant we have what we call cell wall. So in the plant, we have cell wall, and in that cell wall, what of course remember that the cell wall is a rigid part of the cell where it's trying to control or to protect the cell. It provides the rigidity of the cell. So this the cellulose is found in plants and it has the protective functions of the plant cell. And then the next one is exoskeletal insects. Exoskeletal insect of or exoskeleton of insect. Like in mosquito, they are also highly rich in a class of carbohydrate, which also help and protect the mosquitoes from any external attack. So examples of this exoskeleton that are found in insect is chitin. Is chitin. And then the next one is the cell wall of microorganisms. And the cell wall of microorganisms, they are also structural carbohydrates. So we have mucopolysaccharide as a ground substances in higher organisms. So these are actually some basic functions of the carbohydrate. So ladies and gentlemen, if you have questions, you can actually be asking in the comment section, then I will be addressing the questions. So now classification and nomenclature of carbohydrate. So generally carbohydrates are classified into two or into four major groups. It classified into four major groups. We have monosaccharide. So when you said monosaccharide, it means that the molecules having only one actual sugar group. So that is why it is called mono. So these monosaccharides, they are the sugars that can not be further hydrolyzed into smaller units. So it is the most simplest sugar. That is why it is called simple sugar. So these monosaccharides, of course, it has so many examples. We have glucose, fructose, and so many other ones. And then we have disaccharide. So disaccharide is a sugar that contains two monosaccharides. So when two monosaccharides are formed by glycosidic bond, that class of sugar is called disaccharide with the, of course, elimination of water. Okay, just give me 1 minute. So we have disaccharide. So when you said disaccharide, di means two. So when two simple sugars are combined together with elimination of water molecules, then you have disaccharide. And usually these disaccharides, they are formed through a bond. So a bond that joins one monosaccharide with another monosaccharide is called glycosidic bond. So a glycosidic bond is a bond that joins two or more simple sugars together. And then we have oligosaccharide. Oligosaccharide, it means that when you have a sugar of between 2 to 8. So when a sugar of between 2 to 8 are joined or are linked together through glycosidic bond, so that types of sugar is called oligosaccharide. So when you have three sugars, four sugars, five up to eight sugars, when you have them and they join together through glycosidic bond, so are called uh, those types of sugars are called oligosaccharide. And then we have polysaccharide. So polysaccharide is when more than 10 sugars units are linked together. When you have more than 10 simple, or when you have more than 10 sugars joined together through glycosidic bond, so then we have polysaccharide. And of course, even the polysaccharide is divided into two. We have we have what you call homopolysaccharide and heteropolysaccharide, and we are going to look at them one after the other as we are doing the class. So now we are going to start with the simple sugar. We are going to start with the simple sugar. So what is the simple sugar? The simple sugar is the simplest sugar, and it is colorless. It is colorless crystalline solids that are freely soluble in water. So it is colorless, crystalline solid, and it is soluble in water. So most of the simple sugars, they have sweet taste. They are sweet in taste. So, of course, the monosaccharides are those which cannot be hydrolyzed further into simple forms. Also, they are called as simple sugars, and they have a general formula of Cn open bracket H2O close bracket N, and they are subdivided into two. So, carbohydrates or monosaccharide are divided based on the number of carbon atoms, based on the number of carbon atoms, as well as the classification of monosaccharide based on the functional group. Like for example, based on the number of sugars, we can have some sugars that have only two carbon. You can have a simple sugars that have five carbon. Some will have six carbon. So those that have three sugars, they are called they are called trios, yeah, based on that. And we have even pentoses, that is those that have 5 carbon. And of course, based on the functional group, we have aldoses and we have ketoses. So, like for example, this is a classification of monosaccharide based on the number of carbon. So those that have 2 carbon, sorry, 3 carbon, they are called trioses. Right. And then those with 4 sugars, they are called tetroses. Those with 5, they are called pentoses. An example of aldoses or trioses with a 3 sugar, we have glyceraldehyde, and we have ketosis in the case of ketone, those that have ketone functional group. And then we have tetroses, that is those that have 4 carbon. So the sugars that have 5 carbon, they are called pentoses. An example of pentoses aldolase, we have uh, aldoses, we have ribose and xylulose. And then the 5 carbon sugars under ketosis, we have ribulose and xylulose. So these are the examples of the monosaccharide based on their number of sugars, as well as their functional group. So this one, the monosaccharide that are usually found the most abundant in nature, or the most abundant in nature, is glucose. So we have trioses, pentoses, and hexoses. So all these are those sugars that have aldose functional group, and those with ketones functional group, we have something like dihydroses, we have ribulose, and we have fructose. So these are the examples of the sugars that have ketone functional group and aldoses. So those that have ketone functional group, they are called aldoses, and those that have aldehyde functional group, they are called ketoses. So these are the examples of some monosaccharide and their importance, like trioses. Example of trioses, we have glyceraldehydes and dihydroxyacetone phosphate. And the main function of glyceraldehyde and dihydroxyacetone phosphate is A, number one, they are intermediate of glycolysis, and then number two, they are the precursor of glycerol. So you can use these simple sugars to generate glycerol. And then we have tetroses. Example, we have the erythrose, which is actually an intermediate of carbohydrate metabolism. D-ribose, which is on that pentosis, that is those that have 5 carbon. So they have the structural element, or they are form the structural element of nucleic acids, RNA and coenzymes. And then, okay, someone is asking that she didn't get all the example. First, we are starting with monosaccharide. We said monosaccharide is divided into two. It's divided based on the number of carbon. So those that have 3 carbon, they are called trioses, and those that have 4 carbon, they are called tetroses, and those that have 5, they are pentoses, and those that have 6, they are called hexoses. And those that have 7, they are called heptulose. And also, these monosaccharides, they are also further divided based on their functional group. So those that have aldehyde functional group, they are called aldoses, and those that have ketone functional group, they are called ketoses. And these are the examples of some importance, or some monosaccharide that are of biological importance. We have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So the major functions of this is that the glyceraldehyde is actually considered as an intermediate of glycolysis, and then of course dihydroxyacetone phosphate can also serve as a precursor for the glycerol. And then we also have deep glucose, which is the main sugar of the body. We have deep fructose, which converts glucose and utilized by the body. And of course, we have mannose, which involve in the conversion of glycoprotein. Sorry, which serve as a constituent. Okay, the slide with the table. Okay, so this is it. This is the classification of monosaccharide based on the number of carbon. So those that have three carbon, they are called trioses, and examples of them. So under those with three carbon, we have the one that have aldehyde functional group, and examples of that is glyceraldehydes. Then we also have those that have ketone functional group, and they have three carbon. We have it as dihydroxyacetone. So this is the number of carbon. This is the types of the sugar based on the number of carbon, and then based on their functional group, that is aldosis and ketosis. So this is also their classification based on the functional group, that is aldosis and ketosis, and we have example of them. Of course, if you see that, you can see that all of them, they have carbon to oxygen double bond. Carbon to oxygen double bond. So we have D-galactose, which is also another example of monosaccharide, and its main functions is it synthesizes the mammary gland to make the lactose of milk. And then we have mannose, which is a constituent of glycoproteins and glycolipids. So the next thing that we are going to look at is the general properties of monosaccharides. So under this, we are going to look at the general properties of monosaccharides. So example, we have the structure of glucose and fructose, various forms of the isomerism. Under this monosaccharide, we have D and L isomers, we have optical isomers, we have apimers, we have anomers, and then lastly, we have mutarotation. So these are the general properties of monosaccharides. So now we are going to look at the concept of asymmetric carbon. So asymmetric carbon is a carbon that have four different groups attached to it. So when you have carbon, when you have carbon with four different groups attached to it, so that types of carbon is called asymmetric carbon, or in some textbooks you can see it is called chiral carbon, stereogenic carbon, or the asymmetric carbon. All this name is the same for one carbon. That is the carbon that have four different groups attached to it. Like for example here, this is a compound with an asymmetric carbon. So you can see that all the other groups that are attached to this central carbon, all of them, they are different. So that is why they are called asymmetric carbon. And we have a formula of calculating asymmetric carbon, which is 2 power n. So the n stands for the number of functional groups. So if you have two chiral carbon, it means that you have 2 power n. So it means that the number of stereoisomers is equal to 2 power 2. If the number of chiral carbon, if you have two number of chiral carbon, let's say there are two chiral carbon, and then n is equal to 2. So to calculate the number of stereoisomers, we are now going to use 2 power n, which is 2 power 2, and is equal to 4. And then there is no planar of symmetry, and they have mirror image, indicating that they cannot superimpose. It cannot be superimposed. So that is asymmetric carbon. That is a carbon that have four different groups attached to it. Maybe this is the carbon, so maybe you have nitrogen here, you have carbon dioxide here, you have hydrogen here, you have sulfur here. So it means that there are four different groups that attach to the central carbon. So then the next one is furanose and uranose ring. We have furanose and furanose. So the carbon between the aldehyde group of carbon 1 with the alcohol group of carbon 4 or carbon 4, they result in the formation of hemiacetal. So it means that if you have, in the case of aldehyde, there is the carbon 1 in the aldehyde, it has a functional group. So if the carbon 1 of that functional group is joined with the C4 or C5, it results in the formation of hemiacetal. So similarly, the reaction between the keto group of carbon 2 of fructose and the alcohol groups of C5 result in the formation of hemiketal. So the glucose exists in biological system not as a rectangle but as a pyranose ring. Someone is asking, can the slide be clearer than this? I think the problem is from your site. Maybe you have network problem. So in this Furanose and Pyranose cyclization is all about how one carbon in the same molecules can join together to form a cyclic compound or to form a ring compound, which can become in a form of pyranose and furanose. So formation of cyclic structure of glucose. So you can see clearly this is a glucose, and here there is one carbon, and of course here there is carbon 5. So if this carbon 1 and carbon 5 joined together, they will produce a cyclic glucose alpha D glucofuranose. And then of course for the furan, we also have, so in this case they all have anomeric carbon. It means that they have all three carbon, which can be used in the determination of yeah, okay, yes, it can be alpha glucofuranose or beta glucofuranose, which was actually obtained from D glucose. So this is actually how the glucose form a cyclic structure using aldo hexose. And then for the ketohexose, which is the formation of fructose, that is ketohexosis, it is also undergo cyclic cyclization. So in this case, it's a carbon 3 that joined with carbon 5 to generate beta D fructofuranose. So we can so I can get the curriculum of this course so that I can match the first lectures I miss. Okay, no problem. We'll talk about that after the class. So this is how, so there is a possibility that you should understand that the glucose can form a ring structure. So it can form a ring structure, both the glucose with aldehyde group and that of ketone functional group. So that with ketone functional group, it is going to form a fructose. So that is why here we have alpha D fructofuranus, and then we have beta D fructofuranus. And they have all anomeric carbon. All of them, they have anomeric carbon. Okay, someone is asking between what is the difference between alpha and beta. So look at it here. In this structure, you can see clearly here. So look at this here. This one is for the glucose, that is glucofuranus. So in the case of alpha carbon, alpha, look at it, the OH here, the OH here is downwards, is pointing down. While in the case of beta, the OH group is pointing off at carbon 1. That is anomeric carbon. So look at it, in the anomeric carbon, OH group is pointing down in the case of alpha D glucose, and of course, in the case of the beta, the OH group is pointing off. So also here, if you look at it, the OH group in the case of alpha fructofuranus is pointing down, while the beta is up pointing off. So that is the difference between the two. So now, what about the differences between L glucose and D glucose? What is the difference between D glucose and L glucose? So for the L glucose, the OH group is at the left, while in the case of D glucose, the OH group is by the right. We should also try to understand the differences between the two. So the next thing that we are going to look at is yeah, the next thing after this that we are going to look at is the isomers, that is the isometric carbon. Although we explain something like this, so isometric carbon is arise or is actually carbon that have four different groups attached to it. So this leads to the formation of isomers. So this isomers that is formed through the stereogenic carbon or asymmetric carbon is called stereoisomerism. So what is stereoisomerism? Stereoisomerism are actually compounds that have or having the same structural formula but different in spatial configuration and known as stereoisomer. And of course, these stereoisomers have a number of possible stereoisomers depend on the number of isometric carbon. So we have 2 power n. The n stand for the number of stereogenic carbon or the number of asymmetric carbon. Then we also have D and L isomers. So for the D isomers, yeah, the isomers that actually yes, so the D isomers, they have their OH group, or when the OH group in this carbon is on the right, then the sugar is a member of D series. So when it is on the left, then it is a member of the L series. So the distribution of hydrogen and OH group in the other carbon atoms in the molecule is of no importance. Yes. So these are the examples. So if the OH group on the bottom chiral center points to the right, then the sugar is T. So if the OH group on the bottom or on the bottom chiral carbon points to the left, then the sugar is L. So this is what we are going to have at the end. The majority of the monosaccharide occurring in the body are in D series. They are majority in D series. And then we have enantiomers. So optical activity, there is a presence of asymmetric carbon atoms also confer optical activity on the compound. So when a beam of polarized light is passed through a solution exhibiting optimal or optical activity, so it will be rotated to the right or left in accordance with the type of compound. So we have L and D. So the L is standing for laboratory and then D stand for dextrorotatory. So the next one is racemic mixture. Is a racemic mixture. So when equal amount, you see, we say that in D and L isomer. D and L isomers are the examples of stereoisomer. Okay, so Lubaba Yusuf is asking about what is the difference between D and L isomer. So the D isomer, we are just like, for example, look at it here. This is R and this is S. Okay, so now let's look at this compound so we can clearly see the difference between D and L isomer. So we have dextrorotatory and we have laboratory. So first now look at the look at this. Let's just use this methane as an example. So this CH3, it is located at the it is located at the yeah, let me it is located at the right, while the laboratory, in the case of laboratory, the CH3. Okay, yeah, let's just took examples of the one with NH2. Let's say NH2. So you see, they are mirror image. They are mirror image. And of course, in the case of dextrorotatory, you can see that the OH group is by the right, and then in the case of laboratory group, the CH3 or the NH2 is by the left. It is by the left. So that is the difference between laboratory or dextrorotatory. So for the laboratory, the rotating group is actually at the right, while for the laboratory, it is actually at the left. So that is why here, look at the OH group here. Let's just take example of this OH group. So we can clearly here see that the OH group in the case of the lactic acids, it has the OH group is at the it is actually at the right, while the OH group is at the left. So when you have an equal amount of dextrorotatory and laboratory isomers, so the resulting mixture has no optical activity. So if they have 50-50%, so it means that if the number of D isomers and the number of L isomers are the same, then there will be optical activity. And of course, if the number of D isomer is greater than that of the L isomer, then there will be an optical activity. And vice versa, or reverse is the case. So this can apply vice versa. So, but if the resulting mixture has both the same amount of L and D isomer, so it means that there will be no optical activity. So then the next thing is isomers. So ladies and gentlemen, actually I don't want us to take too much time on the lecture. So I think I'm going to stop here, unless if you have question, then you ask. If you have any question, you can ask. So we are going to have our next lecture on Thursday. So you can ask question. If there is any question, you can ask. Is there any question? Yes, of course you can access the slide, Khadija Abdullahi Muye. After this class, After this class, you are going to have the video, so you can access it here. You can access it here in the YouTube channel. Yes, you can access it here. You can always get back to it. So, is there any other question? You see, what I want you to understand actually is now we started going into the really biochemistry. We are starting going deep into the biochemistry. We are now currently we are on carbohydrates, so that is why I don't even want to take too much of my time, so that at least you will have a basic understanding of what it is. First, what I want you to understand is that whenever we say carbohydrates in biochemistry, we define carbohydrate as a fully hydroxyaldehyde or ketones, as a fully hydroxyaldehyde or ketones. So it means that whenever you have a compound that have polyhydroxyaldehydes or that have polyhydroxy ketone, so that compound is considered as an organic compound, sorry, as a carbohydrate. So carbohydrate must either have ketone functional group or aldehyde group with addition of polyhydroxyaldehyde. I will say that this carbohydrate is divided into four. We have monosaccharides, we have disaccharides, we have oligosaccharides, and then finally we have polysaccharides. And we actually define them based on the number of the monomers. So those that have two, they are called disaccharides. And those that have 2 to 8, they are called oligosaccharides. And those that have more than 10, they are called polysaccharides. And then we also look at some kind of isomerism, or we also look at the classification of carbohydrate based on the number of carbon as well as the uh, functional group. So those that have ketone functional group, they are called ketosis, and those that have aldehyde functional group, they are called aldoses. So, ladies and gentlemen, we are going to stop here. So, if you want to access the lecture, you can always get back to here. Thank you very much, and bye. If there is no any questions, then I'm going to end the meeting. Is that all? Okay. So bye.